Biology textbooks often begin with the concept of homeostasis, the idea that the body seeks a static, flat-line baseline to survive. But a living system in motion tells a different story. Survival actually depends on allostasis, the process of achieving stability through constant, predictive change. We remain stable only by continuously shifting our internal parameters to match the environment. The central nervous system acts as the master regulator here, continuously monitoring the body and predicting environmental demands to adjust physiological dials before a crisis occurs. These rapid physiological fluctuations evolve to maximize an organism's probability of survival against acute environmental stressors, like a sudden drop in temperature or a physical threat. While this dynamic response keeps us alive, the mechanical effort of constant adaptation exacts a hidden cumulative systemic toll. This wear and tear is known as allostatic load, the progressive degradation caused by the chronic overactivity or underactivity of our adaptive systems. This macroscopic model shows how predisposing factors, from genetics to early development, feed into our perception of stress. When stressors become chronic, the system fails to return to baseline. This pushes the organism into allostatic overload, flooding the neuroendocrine and immune pathways shown here. Allostatic overload manifests as measurable physical degradation at the cellular level, distinct from simple mental fatigue. These specific tiers of cellular decay are known as the hallmarks of aging, categorized into primary, antagonistic, and integrative layers. Allostatic overload acts as the macroscopic engine that dictates the rate and severity of these microscopic hallmarks. The stress response originates in the brain's control center, the hypothalamus, the moment a threat is perceived. The first pathway to activate is the sympathetic adrenal medullary, or SAM axis, which sends signals directly to the adrenal medulla. This triggers a rapid release of catecholamines, epinephrine and norepinephrine, spiking heart rate and blood pressure to prepare for immediate physical exertion. A second, slower pathway runs in parallel, the hypothalamic pituitary adrenal, or HPA axis. Here, the hypothalamic releases hormones that signal the pituitary gland to trigger the adrenal cortex, forcing the release of glucocorticoids, primarily cortisol. Cortisol drives metabolic changes like gluconeogenesis, preparing the body for prolonged survival by shifting systemic priorities. These axes form a nonlinear network where cortisol suppresses inflammation, and the vagus nerve acts as a parasympathetic brake to inhibit the HPA axis. When chronic stress breaks this reciprocal network, the resulting chemical flood directly attacks the organism's cellular foundation. This damage first appears in the integrative hallmarks, which govern coordination between different tissues and organ systems. Chronic HPA and SAM activation saturates tissues with hormones until cellular receptors lose sensitivity, severing normal endocrine signaling. Biologists define this systemic breakdown as the hallmark of altered intercellular communication. Specifically, continuous cortisol exposure leads to glucocorticoid resistance. The body's primary anti-inflammatory feedback loop is disabled, allowing pro-inflammatory cytokines to multiply unchecked. This results in a smoldering state of immune overactivity known as inflammaging, a chronic low-grade systemic inflammation. This pervasive inflammatory environment alters the gut microbiome and intestinal permeability, driving the hallmark of dysbiosis. The failure of these communication networks isolate individual tissues, leaving them vulnerable to localized metabolic and structural damage. One layer deeper, we find the antagonistic hallmarks, the cell's defensive responses to systemic damage. Mitochondria are highly sensitive to cortisol because they must meet the massive energy requirements demanded by the allostatic stress response. Chronic energy demand overworks the mitochondrial electron transport chain. Jagged reactive oxygen species leak into the cytosol. This structural failure is the hallmark of mitochondrial dysfunction. Severe damage and inflammation force the cell into an irrevocable cell arrest to prevent mutation. The cell becomes rigid and begins excreting its own toxic inflammatory signals, a zombie-like state known as cellular senescence. By forcing cells to constantly overproduce energy, allostatic load burns out the organism's power grid and fills tissues with senescent debris. At the foundational level, we find the primary hallmarks, where the genetic code itself is compromised. These reactive oxygen species, originating from failing mitochondria, act as molecular wrecking balls that aggressively penetrate the cell's nucleus. This chronic oxidative stress inflicts direct lesions in the DNA, driving genomic instability and causing the protective telomere caps at the ends to fray. Cells actively rewrite their genetic expression to adapt to hostile allostatic environments, moving beyond random damage. Changes in DNA methylation and histone modifications silence beneficial genes and lock the genome into a high-alert state, causing epigenetic alterations. Chronic allostatic overload permanently corrupts the organism's source code, reaching far beyond simple superficial wear. Pulling back to the macroscopic level, we can see how this microscopic damage manifests as secondary physiological dysregulation. The accumulation of these 12 hallmarks leads directly to systemic failures in our metabolic and cardiovascular pathways. Clinically, this presents as elevated blood pressure, insulin resistance, and systemic acute phase proteins. These factors deplete our organ reserve, the vital, redundant capacity that allows our organs to handle stress beyond their daily needs. As allostatic load depreciates this reserve, the body loses its homeodynamic flexibility, making even minor environmental stressors potentially catastrophic. The final step of this cascade leads to tertiary outcomes. A fully depleted organ reserve crosses the threshold into irreversible diagnoses: cardiovascular disease, metabolic syndrome, and cognitive decline. The neuroendocrine mechanisms that ensure short-term survival are precisely what destroy the organism over the long term if the system cannot be switched off. The rate at which an organism ages at the cellular level is governed by how its central nervous system processes and adapts to environmental stress.